And I welcome you all for today's talk. It's indeed going to be very interesting because cancer is always a very interesting talk and it affects a lot of people. So we have with us Professor Annapurni Rangarajan, and I'm very happy that she agreed to give the talk, and this is really good that she could find some time. I'll just talk a little bit about her. She has done her bachelor's and master's from Nagpur University, and followed by her PhD TIFR during the same time. She has also worked with Paulo Dotto at the Massachusetts General Hospital, where she has worked, during her PhD also she has worked on, notch signaling, and she continued that. Thereafter, she did a postdoc with Robert Wilbur at Whitehead Institute, MIT, and we did not overlap that time.
Looks like.
Yeah. So anyway, the thing is, it's a great institute within MIT, but a lot of advances happens there, particularly in biology. Thereafter, she joined ISC in 2004 as an assistant professor in the Department of Developmental Biology and Genetics, DBG. She has established a very nice lab with a lot of students always forming a lab. And she predominantly she works on the biology of cancer stem cells and to develop different strategies for tackling cancer related to cancer treatment. She's recipient of a lot of awards. Some of them I will say. Welcome DVT Research Fellowship she is with, and the National Women's Bioscience Award. She's also a member of India, National Academy of Sciences, which is quite prestigious. And I can keep on going, but I will just yes, I will just request Professor Annapurni Rangarajan to enlighten us with her talk. Yeah, please.
Thank you so much. Thank you, Patrick, for the invitation. It's a great pleasure to be here on a day that is raining and cloudy outside. It's nice and warm over here. Is there if we can get this talk uncut? Can you remove that? Because all the titles will otherwise be cut off. By the very top, I can go ahead. So I changed the title as somebody noticed from the abstract, but the content is going to remain the same, so don't worry about it. I'm going to talk to you a little bit in the beginning about basics of cancer biology. I understand many of you are not even probably from biology background. So we'll touch the basics. If you have any doubts, you can ask. We can also sit later and have a chat during lunchtime. But if you feel it's all going beyond you, just tell where is it that we can take a small break and we'll go back and we'll see what needs to be done. I'm going to be talking in less technical terms, so I'm sure you'll be able to be with me. So what is cancer? Where does it arise? Cancer arises in any normal cell of our body. We have so many cells in our body, trillions of them. In any of these cells, we can have a cancer originating. But how does it start? The starting means some changes that need to take place in this normal cell. These changes could be mutations, which means the DNA sequence, the ATGC, the sequence of DNA that can get altered. There may be a mutation which causes a change in the base pair. It could also happen by epigenetic alterations, by which what we mean is there is no change in the DNA base pair, but what changes is the way the genes are expressed. Are 10 copies being made or hundreds of thousands of them being made? That's called a change in gene expression. So any change in gene expression without an underlying change in the DNA sequence is called epigenetics. So such multiples of changes, genetic and epigenetic, when they occur within the normal cell, that's when we get what's called as a tumor. This tumor is in situ, meaning it is in one place. The primary organ in which the change took place, in that organ, the tumor resides. Such a tumor, which is a mass of cells that have now gone wrong, is actually not dangerous to us at all. It may even be there in many of our bodies at this point of time, and our body is well equipped to take care of it. It will go away. Then why do we hear often about cancer being a deadly disease, that it's going to take somebody's life? That's because when this tumor cell learns to move from the primary site to go away far to other organs. For example, if the original tumor is, let's say, in the breast, it may go from the blood vessels and the lymphatics to far away. It can go to brain, it can go to bone, it can go to liver. This is the process called metastasis. The spread of the tumor from the primary organ to a far away different organ is called metastasis. This is what is dangerous to the patient. By the time the tumor metastasizes, that's when oftentimes the patients reach the hospital. Why? Thanks to our government's poor policies, we do not have fantastic screening procedure. If we had early screening, women who's likely to develop breast cancer might detect it much earlier. But how does a woman now get to know that she has breast cancer? Only when it has many times spread. Therefore, by that time, the woman is supposed to be in an advanced stage of cancer. Once the woman hits metastasis, there are hardly any drugs that cure, because with metastasis comes drug resistance. So the doctors also find it very difficult to give a. What in my lab we are interested in studying therefore is, how do these tumor cells, which if they don't know to move away, they are quite okay, but how do they learn to move? During this process, they need to learn to live in an anchorage-independent way. They also need to change their shape from this flat-looking ones to be able to become slender. You know, all women want to be slender. These cells also gain this ability to become slender. What the slenderness gives to them is that it allows them to become motile, moving around, as well as it allows them to invade into the tissue. We'll go through all of these as we touch across all of them. So my laboratory is going to be interested in what are these normal cells in which cancers arise? How do these tumors, which are first located in the one side, how do they manage to learn to move away? Which is when the metastasis takes place. Normal epithelial cells. When we say epithelial, there are three types of cells, or three germ layers of cells. They are endoderm, ectoderm, and mesoderm. That's how when the egg and the sperm fuse and we get a zygote, it tends to give three types of layers of cells. These are called the germ layers. The ectoderm, endoderm, mesoderm. From these three, this entire body, all the cell types come. So amazing. One such cell type is called the epithelial cells. What's so unique about epithelial cell? You look at your skin. This is an epithelial cell. The eye, the outermost, what you can see is all epithelial. What your inner body, that organs are lined up, are also epithelial. You can't see it, but what you can see is the skin. These cells have a property. They are all attached. Attached to what? It's called as the basement membrane. What is that? They themselves secrete extracellular matrix proteins. These are also proteins called collagen and fibronectin laminate. So the cells make these proteins, they secrete it. It's like a Velcro. They have made a bed for them. Now these cells attached to that, they hook up with it. They are also proteins. Proteins called integrins present on the cell hook up with extracellular matrix proteins called collagen, fibronectin, and others. And this is what gives them the attachment. So why is this attachment important? For these cells to get the cue to survive, to make more of themselves, or to make some other cell type. They get all the cue due to this attachment. So what will happen to the cell if it detaches? So attachment is so important. So it's rooted, it's attached. So what should happen to the cell if it is detached? Die. Absolutely. That's exactly what happens. When I take these cells, or when scientists take these cells which are attached, we put them in a dish where they cannot attach anymore. We'll call it a suspension culture. When the cells are suspended in this manner, they die. And this process of cell death is by apoptosis. It's also called anoikis because it's homelessness. The cell which is supposed to be here, now if it detaches, it's going to go away. So in the normal tissue, such a phenomena doesn't occur, but the cancer cells move. So this is what we are interested in studying, how cancer cells acquire this ability to come out, yet stay alive and move. So the only time cells could live in an anchorage-independent way and form these kind of spheroids, which are a mass of cells, a three-dimensional architecture, which divide in the absence of anchorage. Only when there is this tumorigenic transmission. The genetic changes plus epigenetic changes convert the normal cells into this mass of cells called the tumor. Otherwise, what is their fate? It is cell death. This is what I was taught when I was a student like you. I did my PhD, I did my postdoc. This is exactly what I was taught, that normal cells die when they are detached. When they have tumorigenic changes within them, they can form this. All of this was to change just when I was transiting from my postdoc into becoming an independent researcher. What changed? We uncovered hidden within this normal sheet of epithelial cells are a small population of stem progenitor cells. These stem progenitor cells present within the normal tissue, now they can be grown out in the format of a spheroid of this type. What did I tell you earlier? If they are normal cells, they die. That's many of them, they will die. But because of the 1,000 cells, only one survives, scientists could never figure out that such a cell exists. Somebody did this experiment of putting all of these cells in suspension, probably went away for a holiday, and then came back and saw many of the cells that were dead, they were all torn. Some cells survived and they formed these kind of masses. We got to know only around in 1990, 2003, around the time I was transiting, that such cells, which are called normal stem progenitor cells, exist within almost all normal tissue in the body. So what is it? The fundamental property of cancer, which is three-dimensional growth, now can actually be seen within the normal context. Is it possible? Cancer cells learn to grow and divide in a three-dimensional architecture by virtue of their counterparts as normal cells. In other words, can I ask, did these cells originate from those cells? Yes, it's possible. We are going to look at this possibility step by step to see how cancers may actually find their origin in normal stem cell. Why do we need normal stem cells in our body? When there is an injury, our cells refuse, appreciate the skin better. You all see your dead skins go away. But does that mean our skin becomes thinner and thinner as we age? No. There are newer cells that renew and they come. They are replaced. The dead cells are replaced. You can see it in the skin. That's what is happening in the entire body. Wherever there are epithelial cells, the cells that are dead, their lifespan is over, they are cleared. What comes up are newer cells. These newer cells are the stem cells that give rise to the functional cell. So the job of the stem cell in the normal context is to repair our tissue, regenerate, and then ensure that until we live or until we die, we have these cells that can repair. Such cells now, we are thinking maybe the bad ones in which cancers originate. Is it even possible? Because in today's life, people are talking about stem cell therapy. You may have heard about it. If there is an organ which is not working properly, can we get stem cells incorporated into the patient and cure a disease? So in the era where we are talking about stem cell-based therapy, I am talking to you about the dangers of such therapy, which might also become carcinogenic if they are uncontrolled. So let us see what we are talking about. We didn't learn that as the only similarity between cancer cells and normal cells. Whenever we think normal stem cell, a hierarchical picture comes in our mind. What is this hierarchy? There is this stem cell. Every cell divides and gives rise to two daughter cells. Very simple. One cell gives rise to two daughter cells. On the one side, the cell will set aside another one such that it can keep doing this. It's called as a self-renewal. On the other side, the other daughter cell will differentiate and give rise to the functional cells of the tissue. So I am a stem cell. I can make two daughter cells. One of them, I will keep it just like me. It's called self-renewal. So that in future, when there is a need, this stem cell can give rise to more stem cells. On the other hand, the other daughter cell will now divide, differentiate, and form the functional, do the functional job of the tissue. Can we employ the same hierarchical architecture now in the context of cancer? If we do so, this is how the picture will look. The picture will suggest within this thing called cancer, this mass of cell, there are some cells called as the cancer stem cells. They alone can give rise to more of themselves. On the other hand, they make all these cells. And if I were to tell you that today's chemotherapy and radiotherapy is very good at clearing these cells, but why does a cancer patient get a relapse? That's because these kind of cancer stem cells, which do not currently get targeted by chemotherapy and radiotherapy, they stay behind. Because they are so small in number, doctors currently, clinicians do not have a strategy to know how many such cells we have left behind after the therapy is given. What these cells can do, just like what the normal cells do. In the context of a wound or an injury, they'll make more of themselves, they'll repair the tissue. This cell thinks, I've got to repair the tissue. But which tissue? The cancer tissue. So what it could do is, it could divide, it could make more of its own kind, it could kill, it could stay dormant. When we say it could stay dormant, it means it can stay quiet, do nothing for a long time. This is why we see cancer patients that have received therapy. For a while, everything is good. Even after the therapy is stopped, the person is fine. After 5 years, after 6 years, the patient may get a relapse. This relapse could be due to these silently sitting cells called cancer stem cells. So what happened in the era, the beginning of this century is, a lot of understanding and knowledge on stem cell biology got applied to cancer biology. So evolved a field called cancer stem cell biology. So within cancer now, we have small population of cells with stem cell-like property of being able to make more of their own kind by self-renewal, being able to other cell types in the body called as the other cells. We'll call them as bulk in the context of cancer. We'll call them as differentiated cells in the context of normal. So two things I wanted you to remember. A, we had thought that normal cells always die. We got to know that within normal cells, there are stem progenitors that can survive, anchorage independence, and grow and live. We knew that within stem cells, there is a hierarchy of stem cell daughter cells, self-renewal and differentiation, the same hierarchy can now be applied in the context of cancer cells. Okay. So my laboratory, as we were starting out, we chose to use these normal stem progenitor cell-derived mammospheres. Why are they called mammosphere? Mamma is for the mammary gland, which is the breast tissue. I was interested in the cancer from originates in the breast cancer. So we would get cells from the breast tissue. Since they are normal stem progenitor cells, they are able to form these kind of anchorage-independent conjugates. So we asked two questions. The two thoughts that I gave it to you. Is it possible that the breast cancer arises in these normal stem progenitor cells? Point number one. We are going to experiment and see how we can do experiments to address this question. Secondly, anchorage-independent growth is very critical for the cell to move from its primary site to metastasis. Again, I can use the same tissue because this now is growing in an anchorage-independent fashion. So I'm going to kill two birds with one organ structure that I have, which are mammospheres. So let's see what are these mammospheres. How do we make them in the laboratory? How do we even get them? It comes from the breast tissue of a woman. Right? There are many of the girls sitting here. I want normal breast tissue. How many of you want to be volunteers? It's an invasive tissue. It's an invasive procedure. You have to go to the hospital. The doctor will give me a piece of your breast tissue. Will you give? He's very hesitant. Will you give? No, no. So nobody will give. I will also not. Because I'm asking for not a skin. What's the easiest tissue we can get? Blood. You put in a needle and everybody 10 ml blood. If I asked you to be volunteers for my study, I'm sure all of you will say yes to 10 ml of blood. But will you give breast tissue? Nobody will give. Then where will I get my normal breast tissue? What do I need? Normal breast tissue so that I can get normal stem progenitor cells. I have a problem. I don't even have the starting material. But let's see what happens in India. When patient with breast cancer reaches the hospital, if it is detected at an early stage, if the woman has gone at an early stage, a small part of the tissue is now cancerous. The doctor can remove that small part and this is called lumpectomy. Just a small tiny part is removed. The whole breast is saved. But as I said, we do not have a good screening procedure. So the woman hardly ever gets to know she has cancer until it has reached a big, higher stage. At that stage, the doctor advises, instead of removing only this part where the cancer is, they remove the entire breast. This is called mastectomy. So the breast cancer surgical removal of an entire breast is the treatment for a woman with breast cancer. Why? Because we did not detect it earlier. By the time it has reached a bigger size, the doctors prefer that it's better to remove an entire organ. Otherwise, there may be leftover cells that will once again give rise to the tumor. So to avoid that, the entire breast tissue will be removed. So surgically resected breast tissue now, all of the entire breast tissue is not full of cancerous cells. It only stays restricted to one region. So my pathologist collaborator will give me 5 centimeters away from the area of the lesion. She'll cut the tissue and give it to me. Can I consider this normal? Quote unquote normal, because it comes to me from the body of a patient who is a breast cancer. But the lesion is far away. I am getting the cells from this side. So I will definitely do some tests to confirm it's not a cancerous tissue. So that will be the starting material. So I have to collaborate with doctors. For the breast cancer patient, the removal of the cancer is good for the patient. So do you think the patient should object to giving me the removed cancer tissue? It will anyway go to the dustbin, but still the tissue belongs to the woman. That tissue, which has been removed from the body of the breast cancer patient, still belongs to the woman. Therefore, I need ethical permission. From whom all? First and foremost from the patient. Are you okay for me to use that part of the tissue which has been removed? And after whatever is being saved for any detection, everything else will go into the dustbin. Can I use the tissue? I need an ethical approval. For this, there are groups, formal groups. It's not easy to do experiments with human tissue. I need this tissue, so I need to have ethical institutional boards. My institute has an ethical institutional board. It's called the Human Ethics Committee. My institute should approve that whatever I'm proposing, it is worth doing. Once I have that approval, I go to the hospital. I collaborate with Kidwai Memorial Institute of Oncology. The hospital has its own approval. Is it okay that we give this tissue? Is the institute that is asking for this tissue okay? If I ask from IISc, they will have probably no hesitation. Established, reputed institute. But from somewhere else, they don't know. They may say, we don't know what you will do with the tissue. I'm not going to give it to you. So 2 boards, 2 ethical committees will approve my bringing this tissue, which is otherwise a waste. Within that, because I'm talking about stem cells, there is another committee called the Institutional Stem Cell Committee. To do any work with stem cells, you again need approval. Now, ultimately, if I have to put these into an animal model like a mouse to ask whether this will form a tumor, I need approval from them. So you see, when do I get time to do science? That's why I have fantastic students in my lab. Lined up in the last row are some of my students. So I go around approving all this ethical stuff. It all takes time. Goes through multiple rounds of ethical committee approvals. But she's my first student. So happy that she was my first student. If ever in life I have to make one more lab, a new lab, I will definitely ask her to come over. This student will take, she was doing 5 courses at the same time, will go to the hospital, will wait there for the surgery to get over. The surgical tissue comes, she'll take it to the pathologist. This is the group of doctors that can actually cut the tissue in there. She'll wait there. From Kidwai to ISC is 1.5 hours. She'll bring it to my laboratory, go to her hospital room, take shower and come because she feels she's now unclean because she's been to the hospital and everything. The tissue waits until she comes. She will do all these procedures and then she'll come back the next day morning 8 o'clock to see whether she's got cells or what is the other thing we dread when we do all these experiments? Contamination. Bacteria and fungus goes often in this. Why? You must have heard often that somebody in the family went to the hospital and then they got multi-drug resistant bacterial infection. Hospitals are full of these kind of drug resistant bacteria. We also get a lot of contamination. Therefore, we in my lab have to have proper cell culture facility because we are dealing with tissue that comes to us from the hospital. So what do we do once this tissue comes? We first mechanically mince it. It's just like chopping that you would do for all your vegetables. It's just that it happens to be a human breast tissue. Then we include enzymes. These enzymes are good. If everything is tightly bound, we can't get the cells out. So these enzymes remove the cementing substance between the cells. What we get are organoids. These organoids are still multicellular structure. We want single cells like this. We put in some more enzymes. That's when we get single cells. These single cells, we now put it in culture dishes. If they attach, they differentiate. I want stem progenitor cells. So where do I put them? I put them now in newer types of dishes where they cannot attach. So the idea is I take cells from the patient, put them in dishes where they cannot attach. So whoever will grow and give me these kind of outgrowths are stem progenitor cells. Is that enough? Are you convinced these are stem progenitor cells? Is there anything I need to do to convince you? Will you buy my argument? So what do you want me to do?
The cells will die.
Yeah, majority. You can see that these single ones, they are dead. So 99.5% of the cells are dead. 99.9% of the cells are dead. Only these many come up. I'm showing this in a view, you can see so many. But from a starting large number, only these many survive. Are you convinced that these are given by stem progenitor cells? Will you take it for granted because I'm saying so? Science never takes it for granted just because I say so. So what do I have to do? How can I convince you better? Prove? I have to prove they are stem cells, right? The best way to do so is to have some markers. Let's see how we do that. The same cell that came from the patient. On the left-hand side is routine cell culture. The cells go attached to the bottom of the plate. When they do so, they spread out. This is the same starting material that is growing in an adherent attachment culture. The same half, the other half, I put them in suspension culture, and this is what came out. These are floating three-dimensional spheroids. If they are stem progenitor cells, they must be making markers of stem progenitor. So we as molecular biologists do these kind of experiments called qRT-PCR, wherein RNA pertaining to all these names, these are molecules, these are proteins that maintain self-renewal in this tissue. I compare the adherent cell versus these cells are called mammospheres. So ATP stands for attachment, MS stands for mammospheres. I take these two, I crush the cells, and now I'm looking for markers. These are all the markers. These markers of stemness, are they expressed more in my in the mammospheres? That gives me one idea that indeed, compared to this, these structures that have grown in the laboratory have more stem-like entity. In addition, we do marker-based analysis. Some are cell surface markers. We also ask if you are indeed stem cells. If I dissociate, I put you in culture again, will you be able to form? Because you remember they self-renew. If they self-renew, they should be able to make more of their own kind. If they can make more, they should be able to make more spheres. Let's see, can they really make such spheres continuous? That's the patient tissue. That's the first time it made spheres in the laboratory. A week it takes for it to become a sphere like that. Such a sphere will once again put enzymes and make it single cell. Once again, put it in another dish and ask, do these cells have the ability to grow out as a spheroid? Next one, we call them secondary sphere. Another week gone, tertiary sphere. Another week gone, the fourth week, the fourth time it forms sphere. After that, it never forms. What could be wrong? The cells are red, but they do not form spheres. Maybe I lost the stemness property. Maybe they underwent what is called as senescence. Senescence is when cells, as we culture them, they lose a small part of the telomeres. As every time the cells divide, the end of the chromosome, there is a shortening that takes place. If this happens, at some point of time, the shortening will eat up into the coding regions of the genome. These cells will undergo senescence. We measure that by using the staining called senescence-associated beta-galactosidase activity. This enzyme is upregulated in senescence cells. So all blue cells that you see, so organoid, T1, T2, T3, T4. What is happening to the number of blue cells? Increasing. That's what my graph is. So what's happening when I take this 10-cell enriched culture, grow them in culture, over a period of time, they are becoming senescent. One fundamental property of when cells become senescent, the word senescent is also associated with aging. They cannot divide. So senescent cells cannot divide. If they can't divide, they will obviously not be able to form spheres. Now, if they can't divide, can they form cancer? No, because cancer is also defined as a disease of uncontrolled proliferation. So the cells, only 4 passages they grow in culture. After that, they undergo senescence. But our idea was to see, will these stem cells ever become cancerous? So I have to overcome 2 roadblocks, overcome a roadblock of senescence, so that the cells now become immortal. If they are immortal, they can divide, and never become senescent. So this, I'm told many sadhu sannyasis want, or many people in the world also want to become immortal and live long. I'm not one of them. But these cells actually, only if they can overcome this roadblock of senescence, which is a proliferative roadblock, will they ever become immortal. By becoming immortal, subsequent step, can they become cancerous? This is the question that we want to ask. So without any manipulation, without engineering, without doing anything, the fate of these cells was senescent. Now let me see what I can do to convert this cell into these cells. This is where my training in my postdoc lab came up very handy. That was the lab, Dr. Robert Weinberg's lab, which had shown you can take any normal cell, many normal cells, convert them into immortalized and transformed cells by doing a simple trick. What is the trick? You introduce 3 genetic elements into the cells. What are these elements? One is a viral protein. It's called the SV40 early region. Another is HTERT, which is the catalytic domain of the telomerase enzyme that allows you to prevent the reduction of the telomere gland. Another one is an oncogene. What do these things do? The roadblocks present within our cells, which prevent actual tumor formation, are called tumor suppressors. They are there in our body. What this protein does is it inhibits the tumor suppressors. That is, one hit, I'm going to get rid of multiple tumor suppressors by putting in this element into the cell. On the other hand, RAS is an oncoprotein, which allows the cells to overcome cell death. It allows the cells to proliferate. So on the one hand, tumor suppressor break I'm removing. On the other hand, I've got oncogenes which are going to tell the cell, divide, divide, divide. There's no need to stop. Even if the cell, the entire genome is not duplicated, even if there are trouble, divide. What is the telomerase maintenance going to do? It's going to tell the cell, even though you are dividing multiple times, your ends are not going to be depleted. Therefore, this combination of 1, 2, and 3 is sufficient to convert a normal cell into a cancerous cell. It was a landmark discovery. By putting in knowledge of 20, 30 years, the laboratory had actually managed to convert normal cells into cancerous cells. We asked, can we employ this strategy now? But to which cells? The cells that we are growing in our laboratory, which are the mammospheres. These stem progenitor enriched cells, can I now apply the same strategy of including 1, 2, and 3 genetic elements and ask, will these cells now become cancerous? Is it even possible to convert a normal stem cell into a cancer cell? So what do? We have tools of viruses. Viruses are not just infectious agents. Scientists have tamed the viruses, and we can use virus to introduce these elements into the cell. We'll not go into the details of it. So we took these normal mammospheres, stem progenitor enriched. One by one, we started introducing SV40 largely, the HTERT. After introducing these two, we actually asked one question. We know our cells are dotted because they are glowing green. GFT comes from these constructs, and therefore I know that my cells have actually received the construct. After putting the first two, we waited and we asked a question. The job of this third protein, oncogenic RAS, is actually to enable the cancer cell to live in an anchorage-independent condition, to grow and form spheroids. But our starting population itself is anchorage-independent. So is it possible that these cells don't even need RAS if they are starting as a stem cell? So we asked, why wait? We've got these two oncoproteins introduced into the cell. Let us now test whether they are tumorigenic. What's the best way to do this? I can take an immunocompromised animal, which is normally a mouse which is immunocompromised. Why should it be immunocompromised? Because these are human cells. And I'm putting them into mouse. So what will the mouse do otherwise? Can I take your cell and put it into them? What will your body do? You must have learned. What does it do? It kills. It's called host versus graft rejection, right? We cannot get kidney from anybody. We have to have the closest relative. Then only we can do a kidney transplantation. These are human cells. So obviously, when I put it into a normal mouse, the mouse will reject all of this. So scientists have developed what are called as immunocompromised animals. In these animals, the not-skip mice, the immune system is very poor. Therefore, in these immunocompromised mice, if I put the human cell, it will not reject these human cells. See, scientists are amazing, right? They have all these cool techniques and tricks to play. So what happened? I've got the two of these components. I put them into these cells, injected them into an animal, and both the poor animal actually formed a tumor. And this tumor, when we take it out, this is how it looks like. So it's a mass of cells. It's a tumor that has been formed by the normal stem progenitor cells, into which biologists engineer these two constructs. Biologists can at times be engineers, okay? So the bioengineering that happened in there allowed the normal stem progenitor cells to now express the sp40 large T genomic region as well as the telomerase, the catalytic domain of telomerase. So only two components was enough to convert these cells into tumors and form these. Now somebody might ask, how do you know this is coming out of the cells that you injected? So we take this tumor, we make sections of these, and we prove once again that indeed the proteins that I put in it, they're actually present. This is how science goes by. Every single thing that you do, you have to prove it. And we often use markers. So these cells are green. They came from the retroviruses. They also express a protein called RAS3, which came out of this construct. So we are convinced now that the tumor that is formed here is because of the cells that we injected into the mice. And remember, I talked to you about cancer stem cells. These are subpopulation of cells within a tumor, which alone have the ability to give rise to new tumor. Just like normal stem cells alone can give rise to a normal tissue, likewise, the subpopulation of cancer stem cells alone can give rise to new cancer. Do the cells that we made, which is the NBLE cells, do they also have cancer stem cell? How do we recognize this? Cancer stem cells can be recognized by two markers they make, CD44 and CD24. It's just like, how do I recognize you? You wear glasses, you don't wear glasses, you are this height. It's the phenotype. This is what is called phenotype. Likewise, every cell on its surface has proteins. These antigens are different from one cell to the other cell. I can put an antibody now that recognizes a particular antigen. Doing that technique and using a machine called flow cytometer, where one by one these cells will go. And if there are two colors, I can sort them out into 4 quadrants. Both color positive, both one high, one low. There are 4 possible combinations, and you can have 2 different markers. Out of these, we knew that only the CD44 high, CD24 low, or the cells belonging to this quadrant are known to be cancer stem cells. But how do I figure this out? We isolated cells from this quadrant, we isolated cells from this quadrant, and asked independently which of these quadrants have the ability to A, form these kind of spheres, and B, form a tumor in an animal. Only those cells that can do this are called as the cancer cells. And it turned out that only cells belonging to this top quadrant were able to form spheres. They were the only cells that were able to form tumors in mice. The other cells, they did not form these kind of spheres, did not form this. They are all cancer cells. But within cancer cells now, only the subpopulation of cancer stem cells have the ability to in vitro form mammospheres and in vivo give rise to tumors. That's why if the chemotherapy now gets rid of all the other cells, but leaves behind these cancer stem cells, they are the ones that are likely to give rise to a new tumor. So what have we done? We started from normal stem cells, engineered into it 2 constructs, which gave rise to NDLE, laboratory engineered stem cell derived cells. What is the nature of the tumor that they form? They ended up forming, technically these are called as invasive ductal adenocarcinoma. Forget about the mouthful of the term. 85% of the cases that the clinician will see in the hospital are of this kind of things. What about those which previous researchers had done? I had learned this technique in another lab. What about the experiments they had done? When they made this, they had used self-pepper adherent. If the adherent cells took up all of this, they made types of cancer that only represented 2 to 5%. But when I started off with stem cells, did the same gimmick, did the same experiment, we got the kind of tumor that is 85% seen by the clinic. Can we now make certain assumptions based on this? Is it possible that many women that get these kind of cancer, actually the origin of that is from the normal cell and normal stem progenitor cell? We cannot confirmedly say it, but the threat is there that normal stem progenitor cells can become cancerous, and such cancers can then give rise to these kind of the types of cancer that the clinicians encounter. So I want you to take this as a message. All they have shown is that normal stem progenitors have in them the ability to become cancerous. This is not to say that we have proven that all cancers originate in stem cells. At least some cancers originate in stem cells. That's what I want you to take it as a take-home message. We'll now go to another step. I said my lab was interested in two aspects. One, which cells the cancer arises. Two, how does the tumor learn and acquire the ability to metastasize and move? We are again going to use this mammosphere. Why? Because it's an anchorage-independent entity. Normal cells are attached, but they have to detach and they have to live. So how does anchorage independence happen in these tissues? Of course, I want to know about it in the cancer, but then if I can use this normal structure, if I can figure out the molecular mechanism within the structure, maybe cancer cells also use the same. So let us see, how does this normal mammosphere ever live in order to form this kind of an anchorage-independent structure? We all experience stress at different time points. When there is an exam, you experience stress. When you have to catch a flight or a train. These cells that grow in an anchorage-independent fashion, they are also stressed. Why? Because A, they are anchorage-independent. And these are epithelial cells. They love to attach. They need attachment. Are deprived, so they are stressed. They also, there is hypoxia, less oxygen in the center of the core. Because oxygen diffuses. And from outside to inside, there is a little less oxygen. Nutrient also, similarly, there is a little lack of nutrients inside. So the cells here are actually experiencing multiple types of stresses. So how do we start about asking what could be the molecular mechanism that could enable these cells to live and overcome these kind of stresses? So we got interested in a protein, very interesting. It's called AMP activated protein kinase. You all know what AMP is and ATP is. You might know what is ATP. It's called price. What does it do in the cell? Energy. Currency of energy in our body is ATP. When ATP is utilized, what is left behind? AMP. That's triphosphate. It's the phosphate bonds that give the energy. ATP, ADP, and AMP. AMP is what is left behind after we have used up the energy. When does this protein get activated? When AMP levels are more. So is this cell in a high energy status or low energy status? Low energy? That's one. So when the cell is in a low energy status, AMP levels are more. AMP goes and physically binds to this protein. The moment it binds, it brings about an allosteric conformational change in the protein such that on top there can be a phosphorylation. Basically, this protein called AMPK gets activated whenever a cell is in an energy low cell, deprived status. In a low energy status, you must have heard about two metabolic reactions. One is anabolic, one is catabolic. Have you heard about it? What is the building up process called? When we build up big molecules. Anabolic. When we break down molecules to get energy, it's called catabolism. Metabolism is anabolism plus catabolism. We build up when we have more material, more energy. So what should a cell that is facing energy deprivation do? Build or break? Breaking down gives you energy. Building up requires energy. Now tell me. It should break. So it should basically break down. Once AMP is activated, it's like a switch. It prevents all, it's like a metabolic switch. It prevents all building up processes. It turns on all breakdown, breakdown, breakdown. Why? Energy is required until actually the value. We'll leave it aside. We asked, If AMPK is a protein which is also activated hypoxia, one paper told us that. In our own lab, we figured out that under nutrient deprivation, when we remove glucose, the cells activate AMPK. So we knew that at least two stresses, stress stimuli, which activate AMPK, hypoxia and nutrient deprivation, is present in this. We asked, is it possible that the same AMPK can also help cells overcome matrix deprivation, anchorage independence? Since the structure is also a stem progenitor engaged one, it becomes a dual query. If I can form a mammosphere, there must be stemness. If I can form a mammosphere, I'm obviously alive. Can AMPK assist these cells to become mammospheres? That is, prevent them from dying when they are matrix deprived and also help in maintaining their stemness property. Let us see how do we go about addressing this. Same system. We got the primary patient-derived normal mammary cells. Put them in adherent culture, they look like this. Grew them as mammospheres, they look like this. Between these two, I'm querying if AMPK is a molecule that is activated. How do we test for activation of a protein? We can ask whether AMPK itself gets phosphorylated, because that is when it gets activated. It is a kinase. The K stands for kinase. What do we mean by kinase? It can phosphorylate other substrates. So its two substrates are these two. The names may sound very weird. One is called Raptor, one is called ACC. What do we mean by phosphorylation? Adds a phosphomoiety. These moieties come from ATPG, so it transfers. So if AMPK is active, it's going to be phosphorylated. If it is active, these two molecules will also get phosphorylated. We have cool tools in our laboratories which allow us to detect whether, compared to this, do these cells have higher levels of phospho-AMPK, phosphoraptor, and phospho-ACC. What do we use? We use antibodies that are specific either to this phosphomoiety, or it can recognize the total protein which is not phosphorylated. This arrow, and this arrow tells you, compared to the adherent condition, these cells, the mammospheres have elevated levels of phospho-AMPK, meaning AMPK is activated. It also has higher levels of phospho-ACC and raptor, its two substrates. So together, what have we proven? That compared to adherent condition, when cells are matrix deprived, this protein called AMPK gets activated. Its primary job is to be a metabolic switch. We know it is activated. Does that mean it is necessary for the formation of the spheres? It's activated. Does that mean it's necessary? How will you test whether it is necessary? If it is necessary, what should I do to test it is necessary? I can inhibit it. What else? Knockout it. So she's my student. So she's a biologist. She has better tools than inhibitor. We can either use inhibitors to inhibit the protein, or we can simply remove the gene itself. Okay. Now let us see what happens. If we use an inhibitor, there is no formation of mammosphere. If we use AMPK silencing, removal is called silencing. I don't have any more AMPK made. I don't get any mammosphere formation. So not only is AMPK activated, but it is required for the formation of mammosphere. Right? All set. AMPK gets activated. It is indeed required for the formation of mammospheres. Then we ask the question, how is AMPK doing? Doing what? How is it preventing the cells from undergoing cell death by apoptosis or finite death? I will cut these stories short because you guys are from varied background. So as cell biologists and biochemists, we have a lot of tools. We identified PA15 as a new molecule, which is downstream of AMPK. And we figured out that AMPK phosphorylates PA15. And this phosphorylation is what now allows the cells to prevent apoptosis. How does apoptosis take place? There are death ligands which bind to death receptor. Inside, these molecules form what is called as a death-induced silencing complex. Weird names, take it. Together, they cause apoptosis. Now, what happens when AMPK phosphorylates is that it goes and binds to this disc complex and actually prevents the formation of this disc complex. By preventing the formation of that, it prevents apoptosis. So very simple. AMPK gets phosphorylated. It phosphorylates another protein, thereby preventing the formation of a complex that is required for undergoing apoptosis. So what did we uncover as part of this? In normal mammospheres, normal stem progenitor cells, matrix deprivation causes AMPK activation, which phosphorylates PA15 and thereby prevents apoptosis. But these are all in normal cells. Our idea was to know, can I learn from normal cells and see if this applies to cancer cells? Now I went and I will completely cut the story short. Exactly all the experiments we did with normal cells, we now did with cancer cells. Which cancer cell? Remember, my original tissue comes from a breast cancer patient. I get the cancer cells also from the patient. So the cancer cells that I got from the patient also activate AMPK and show the same thing. So what are the three things I learned from here? AMPK, which people, a lot of times we as scientists are tuned in our mind. Anything that promotes proliferation is oncogenic. Anything that prevents proliferation is tumor suppressor. But to begin with, I told that AMPK suppresses because it inhibits tumor. But now what am I showing? It actually allows the cells to become survive. So it's a pro-tumorigenic molecule. Again, does this support? Because the same stem cell, these cells have the same pathway AMPK P13, so does the cancer cell has. Can we say it supports the stem cell origin of cancer? So multiple experiments, part by part that we do, seems to suggest that indeed cancer cells may have learned to live in an anchorage-independent state by virtue of probably originating from a stem-like cell itself. Okay. Now, over a period of time, my laboratory, 10 years down the line, multiple students joined the laboratory. So we first showed that AMPK plays a role in anchorage-independent survival. Different terms that I have not talked to you today, but you remember metastasis. You remember I said becoming slender is important to become metastatic. AMPK seems to be central to all these multiple mechanisms, all of which, what do? They promote metastasis. They promote spread of tumor. So you will agree with me that AMPK is bad for the human being. It's good for the cancer because the cancer is spreading. But what should the clinician do? What should the doctor do to treat the cancer patient? Suppress AMPK, inhibit AMPK. But if I told you that the clinic is looking at it in a completely different way, and I'll tell you why. AMPK to begin with was called a tumor suppressor. Why? Because it inhibits proliferation. Okay, inhibits proliferation, it's a tumor suppressor. What did experiments from my laboratory over the past 20 years show? AMPK is central to multiple places which allow the cancer cells to metastasize. So AMPK is critical for metastasis, whereas it may be inhibiting proliferation. So it's okay at times to not proliferate, but live. If you don't live, you cannot proliferate. So sometimes it's okay to say, I will not proliferate. It's a trade-off at the expense of proliferation, but I'm going to live. I need to see another day to be able to proliferate. So what's this? What we have done is that we have identified AMPK overcoming stresses faced by cancer cell, such that we are now beginning to call it as a tumor promoter. And also it's a contextual oncogene because only in the context of cells, when they are experiencing stress, does AMPK actually come into picture, activate, and then overcome stress so that the cells can survive. Otherwise, it remains as an inhibitor of proliferation. So now, what is it that became very intriguing is that while, like you, we all proposed that AMPK inhibition is a novel cancer therapeutics, correct? But this is not what the field was doing. Once again, recall, what does AMPK do? It is good for the tumor progression, correct? There's a drug called metformin. Have you all heard about this? Metformin is the #1 anti-diabetic drug. Anybody who gets diabetes, the first level treatment is metformin. People in the field of cancer biology were repurposing and they figured out metformin inhibits cell proliferation. Turns out that women that were taking metformin for their diabetes had lesser frequency of getting cancer than the women that were not taking metformin. So it said that metformin is actually good for the patient. If we activate metformin, there should be less number of tumors. But the tragedy is this, that metformin is an activator of AMPK. The job of metformin, the way it works as an anti-diabetic drug, is by activating AMPK. What did I tell you about AMPK? AMPK plays a crucial role in the spread of tumor. So are you thinking the right question? What is the right strategy in the clinic? To inhibit AMPK or to activate AMPK? Inhibit. Unfortunately, because metformin is a cheap drug, and there was longitudinal data saying that women that took metformin had less frequency of getting cancer, and metformin, by activating AMPK, was inhibiting tumor proliferation. This went into clinical trials. Zillions of clinical trials came up. And surprisingly, not to us, but to the rest of the world, there was, so, but okay. Actually, many of the clinical trials had absolutely no benefit. Some of them even said that the people had poor prognosis, meaning there was more metastasis. This is what we would have predicted. But the world, we were having two kinds of blocks. One was a roadblock. I'll tell you about it. This was a mental block. The field for 35 years had thought about AMPK as an anti-tumorigenic agent because it's an anti-proliferative agent. Therefore, even today, when our papers go or we write grants, there will be somebody sitting there who will say, but AMPK activation is what the strategy should be in the clinic. But what are we trying to tell? That it's a contextual oncogene. Whenever there is stress, AMPK gets activated. Activated AMPK promotes tumor progression. Therefore, we need to inhibit AMPK. What is a roadblock for this? There is no FDA-approved clinically available drug. So we do not have an inhibitor that we can straight away take to the clinic. We have small molecule inhibitors. These are called pharmacological inhibitor. We can use it in cell culture. Occasionally, we can put it into an animal for a short period of time. But nobody has come to the level where we can actually apply them into the patient. So we do not have specific AMPK inhibitors. That's why at the end of my abstract, I said there's an urgent need for people from multiple fields. It could be chemistry, it could be organic chemistry, natural chemistry, to come and identify drugs because there is evidence which is very strong at this point of time to say that AMPK inhibition is what the tumor therapy should be all about. I will end my talk with this and not go to the last part because I think it's already time, right, Partha? I think I've talked about two different stories. I had animal model, but I'll end this. I'll quickly go to the very last slide because a talk cannot end without acknowledging the batches of students that come to my lab. Partha was very surprised to see that I have always a big number of lab, a big size lab, and that's indeed true because we have to go, somebody has to go bring the tissue, somebody has to process, somebody has to do the experiment, analysis. 5 to 6 years is never enough. So somebody finds a good observation, the next generation actually follows it up. So that's all my bunch of students, always smiling, always happy. Some of them are new, so not yet smiling that much. And I should thank all the patients that donate their samples to us. The doctors that have been fantastic collaborators in giving us tissue, many collaborators, and no experiments get done without funding agency. So the whole lot of funding agency for their money. And thanks for your time and attention.
Yeah. Now we know more about cancer. Good, good. I'm really happy. Thanks a lot. It's really great to get into all the details of
what happens within a
cancer. Yes. I mean, everybody is some or the other way, knows about this cancer. They just encounter it somewhere. Okay, so anyone want to ask, have any questions? Yeah, please go ahead.
Ma'am, can we apply this technology in other cancers like blood cancer?
Yes. In fact, the origin of this theory of cancer stem cells came from leukemia. Absolutely. So the same, we applied the leukemia strategy into breast cancer. So the cancer stem cell concept now appears to be holding true for multiple cancers, not all. That's a different class of story. I did not talk to you about it at all. DNA repair, whenever there is a DNA damage, we want to prevent that. Yes, DNA repair agents are there. Whether cancer stem cells are ablated by it, no. But that's a different strategy, different thought. Anybody else?
Anyone else? Okay, if not, I'm just trying to understand this. I have some questions.
Oh, sure.
I mean, what is the trigger for these normal cells to turn into cancer cells? What is that?
We cannot say convincingly from this data that indeed it is a normal cell that becomes cancerous. It's circumstantial evidences that are telling us. Again, the trigger is a genetic change, which could be a carcinogen that comes from outside, or a carcinogen that is produced within our body by the metabolic. It could be multiple such changes that take place from outside and inside, but happening within the normal cells. Whether it happens in the differentiated cell or the stem cell, they're all coming from outside, DNA lesions, epigenetic alteration.
Right. So can we say that the way, we live now as compared to 30 to 40 years back, modernization,
yes,
does that have a effect on this? We're using more plastic, we're using more other things like that, maybe our body.
Absolutely. The graph of incidence of cancer does show that compared to 30 years, we have more, but that could also be due to more awareness, more people reaching the hospitals. But if you ask me, will our lifestyle change affect cancer? It is a known factor that lifestyle changes affect. And I put it in this way. To begin with, breast cancer was not the #1 cancer in India. It was cervical cancer. And then it turned out that 4 metropolitan cities Bombay, Chennai, Delhi, there the incidence of breast cancer was more there than in other villages and small towns. So what was the cause? Completely lifestyle change. The woman's outlook in the metropolitan city, the food we eat in the metropolitan city, delayed childbirth, because the woman in the village would have childbirth between the age of 18 to 24, whereas in the city it's going to be 24 and above. So this delayed childbirth with its hormonal influences is also one cause. So lifestyle changes is definitely something that can affect cancer.
Okay, there's something that I thought may not be right, but I'll ask. Autophagy is a way to tackle cancer.
One way by which AMPK allows the cells to withstand stress is by promoting autophagy. So autophagy can be bad and good. Prolonged autophagy, when the cell eats up its own organelles, prolonged time, it's bad. Because obviously I've eaten up a little bit of it. But until the time that there is stress, like there is a famine, until that time, if you can manage to eat up a little bit of your organelles, AMPK does exactly that, promotes that amount of autophagy, which allows and gives energy for the cell to survive. For a very long time, if the other good things don't come, maybe the cell will die. But the blood vessels will come, and in future there will be more nutrient available. Until then, AMPK promotes autophagy. A bit of eating yourself is good.
So you're indirectly suggesting that people having cancer can go for fasting.
Absolutely.
And that helps.
Fasting, I would look at it in a different way also. Now, when we think autophagy, you remember I've never fasted in life because I need food. So my grandma did it, my other relatives did it. Once a week they'll fast. What Shivratri they will fast, Amavasya they'll fast. Perhaps that's a good way to remove all the bad organelles in our body. Once in a while, if you fast, the accumulation of the bad organelles, maybe they get eaten up. Autophagy also talks about the epigenetic aspect that I didn't talk to you about, changes in gene expression without changing base pair. No mutation, but you change gene expression. The way chemotherapy can be given, there have been trials that said if a patient is fasted overnight and then given chemotherapy, apparently that is more effective than capital. So that is fasting. So fasting probably allows different gene expression to come up and probably opens up these cancer stem cells and make them amenable for the chemotherapy. So I would say interim autophagy, interim fasting is good.
So we have some solutions. Okay, anyone else want to ask anything?
No question can be stupid. Remember, only answers can be stupid.
Very tight bra is reason for breast cancer.
I would not think so. It's not reason. But maybe for a very long time, restriction, it's an organ that's also growing every time. Every time a woman undergoes menses, the mammary gland actually expands a little. Right? So that means we are constantly keeping it compressed. I wouldn't necessarily think that it is a cause for cancer.
Devising AMPK inhibitor can help in preventing cancer.
Say that again. What is the first?
First, devising.
Devising. I agree. So we have this one chemical called compound C, which is pharmacological inhibitor. We took animals, we made tumors in them, and we gave AMPK inhibitor compound C, and the tumor shrunk. And along with it, when we gave chemotherapy, we needed less chemotherapy to kill the cells, kill the tumor. We also realized that once AMPK is inhibited, I said the leftover after the therapy, oftentimes the cancer stem cells are left over. There were less number of cancer stem cells left over after. I did not go into those many details. So that's why we are advocating now. AMPK inhibition could come up as a good cure for this. It could be across multiple cancers. All epithelial cells have AMPK. Many epithelial cells activate AMPK when it's matrix. So it could apply. We chose to study breast cancer. It could apply for other cancers as well. But there is no inhibitor right now. Till now, because there are only few. I showed that fish going upstream. The struggle for us has been really going upstream against the wave of everybody else, the mental block, everybody else thinking metformin, activating AMPK is good for cancer treatment. Clinical trials, one after the other is going on. After 10 years now, we are getting the outcome of some of these clinical trials. At least absolutely no effect or worse. This is what we expect if you activate AMPK in a patient who already has cancer. It's likely to have more dormant cell, likely to come back with cancer, or that's what our model suggests.
Right. I think we have quite a number of questions, so
We
we
can
can
make
discuss. Yeah, but thanks a lot.
Thank you
It's
so
a
much.
wonderful talk.
Pull out